Joining us for today's European Pharmaceutical Review webinar titled The Future of Sterility Testing, One-Day Results with Advanced RT Real-Time PCR Technology. My name is Dr. Carrie Haslam, editor, and I'll be moderating today's session. On behalf of European Pharmaceutical Review and Fujifilm, I'd like to thank you all for attending today. So I'm delighted to be joined by Dr. Julia Matros, Associate Sales Manager at Fujifilm Waco Europe GmbH, who will discuss the latest advancements in sterility testing, focusing on how RNA-based detection and RT real-time PCR technology is transforming the field. Julia will also explain how this approach enables reliable one-day results, enhances sensitivity and detection, and integrates into existing workflows. Following the presentation, we'll move on to our Q&A session, so please feel free to submit your questions at any time during the discussion via the questions panel on the right-hand menu. And with that, I'll hand over to our expert, Julia, to begin the webinar. So over to you.
Good afternoon, everyone, and welcome to today's webinar with the title, The Future of Sterility Testing, One-Day Results with Advanced Reverse Transcription Real-Time PCR Technology. My name is Julia Matros, and I'm working for Fujifilm Vaco Chemicals Europe. I'm very delighted to have you all here in our webinar today. This agenda outlines the key topics of today's webinar. We will start with a short introduction regarding the rapid sterility testing before we will have a look to the sterility testing in general in the pharmacopoeia. After that, I will show you some key points to our product, the RiboNut. So what is the source of uniqueness of our product and the outline and flow of our assay. After that, we will have a look to some experimental data and some validation data. But let's start with the introduction to rapid sterility testing. So what is sterility testing in general? It is a critical quality control measure in the pharmaceutical industry, especially the QC department, to ensure that medicinal products are free from viable microorganisms. It serves as one of the final verification steps to guarantee patient safety, biosafety, and product efficacy, especially for sterile products such as injectables and cell-based therapies. Biosafety in this case involves safe practices for handling biological materials, especially infectious agents. These infectious agents include pyrogens, microorganisms, and mycoplasmas. So let's have a more detailed look. This slide here shows the moves and trends in the pharmaceutical biosafety testing. You can see different contaminants categorized into different categories, and then the substance to be measured for each contaminant, the official method, the main driver to change the process, and the progress in the change. So let's start with the pyrogens. The substance to be measured regarding pyrogens are endotoxins. The official method currently to measure it is the LRL test. The main driver to change to the recombinant reagent test in this case is the sustainability. On the other side, you have non-endotoxin pyrogens, also called NEPs. They are currently tested with the rabbit pyrogen test, and because of the animal welfare, there's a change to the monocyte activation test. Then we have microorganisms, for example, bacteria and fungi. The current method for testing it is the cultural method, and the need for speed shows that we need a rapid sterility test. I will come later on more onto this topic. Then we have the mycoplasma. The mycoplasma is also tested with a cultural method, and here's also the need for speed, the main driver to change to a rapid test. For our webinar today, the microorganisms are very important. Here you can see different sterility testing methods that are currently on the market or be used. You can see them with their characteristics and their performance. For example, you have at first the compendial method, which is a culture method. It takes up to 14 days, or it takes 14 days. The sensitivity is very good with 1 to 3 colony forming units, and you can test also cellular samples with the culture method. In the last column, you can see whether it is able to detect dead or only viable microorganisms. Below the compendium method, you can see different other methods, for example, the respiration, the ATP detection, or the solid phase cytometry. The NAT method follows under all the mentioned methods before, and the NAT method is a new method on the market. The time to result here is 4 hours, but the sensitivity, as you can see it in the table, is not so good with 10 to 100 CFU. So what we can see here is that we developed a new product with the name Ribonat, which takes up to 7 hours, but has a sensitivity below 9 CFU per ml. So NAT in general is the nucleic acid amplification test. As I explained before, there are some advantages. It is super rapid, it is applicable to cellular samples, and you can use the general purpose equipment, but there are also some disadvantages, for example, the lower sensitivity with the 10 to 100 CFU and the risk of false positives by the residual DNA. As I said, we developed the Ribonat product, and the Ribonat product utilizes the advantages of the normal NAT method while reducing the drawbacks. We will come later on more in this topic in more detail. So let's come to the sterility testing in the pharmacopoeia. What you can see here is on the left side the pharmacopoeia, what we are talking about, then the important sterility test chapter in the pharmacopoeia, and the information for alternative methods. Rapid sterility testing is currently still a promising alternative method, but as I said, it is an alternative method. So important for you or for all company working in the sterility testing fields, of course, you have at first a look to the sterility test in general, the normal chapter or the compendial chapter, but you can have also a look to the alternative methods. In addition, there are also other guidelines, for example, the ISO norm 24190 with the name Biotechnology Analytical Methods, where you can find more information regarding alternative methods, and the PDA Technical Report number 33. So the compendial methods are for the USP, the EP, and the Japanese pharmacopoeia harmonized. But what we have to say here is all of them require a 14 days culture. You have to imagine that certain cell-based medicinal products have very limited shelf lives, ranging typically from 2 to 3 days. We have it also here in the timeline shown. So sometimes you have these products with the limited shelf life. Normally, they have to be administered to the patient at day 2, but the sterility test result is available on day 14. So we can say there's really a critical need for rapid sterility testing methods on the market. So let's come now to our product, the Ribonat, and the source of uniqueness of our product. So at first, I want to show you some key features of our product. We developed a one-day rapid sterility test, which takes approximately 7 hours. And our test is detecting the ribosomal RNA using the reverse transcription real-time PCR method. It has a higher sensitivity than the genome detection with a concentration of 9 CFU per ml. Ribonat reduces false positives from dead microorganisms and from the residual DNA. And it has a wide detection range of bacteria, aerobic and anaerobic ones, and fungi in one single assay. For our assay, you need 3 different kits: the RNA isolation kit 1, the RNA isolation kit 2, and the detection kit. And what you can see here is that every kit is stored under different conditions depending on the specific properties to ensure the optimal stability and the performance of our assay. Our kit has 3 important steps. The first step is the pre-treatment. It takes 3.5 hours. You have there the activation of the microorganisms and the inactivation of residual DNA. In the second step, which takes 1.5 hours, you have the RNA isolation. It means you have the lysis of the microorganisms and the RNA extraction and purification. And in the last step, you have the measurement step. This takes 1.2 hours and is for the RNA detection with reverse transcription real-time PCR at the end. And these steps together are enabled to fast and reliable stability test. You need for each step different components from different kits. You can see it here. For example, for the pre-treatment in step 1, you need an RNA isolation kit 1. For step 2, you need parts of all the 3 kits we have, and for the step 3, you need the isolation kit 1 and the detection kit. So let's come to the points for the source of uniqueness. Our first point is that we are targeting ribosomal RNA. The second point is we have a special primer and proof design. Then we have an optimized lysis method. We have a pre-incubation step included in our kit, and we have a reduction of false positives. So let's have a more detailed look to each point of uniqueness in the following slides. So at first, I want to start with targeting the ribosomal RNA. On the left side, you can see the conventional NAT method targeting the genome DNA. On the right side, you can see our product Ribonat targeting the ribosomal RNA. In the table below, different strain names are listed and the copy number per CFU, once for the genome DNA and once for the ribosomal RNA. We can say here from this slide that the ribosomal RNA amount is higher than the target copy number of the genome DNA. That means smaller numbers of microorganisms can be detected. The second point is our special primer and proof design. At first, we have a high sensitivity, and then we have also a wide coverage and specificity. Let's come to the high sensitivity at first. So we are targeting the large ribosomal RNA, as I said before. That means it is not affected by the secondary structure of the RNA. Then you can find in many different bacteria conserved sequences. For bacteria, it is, for example, 16S rRNA and 23S rRNA, and for fungi, 18S rRNA and 25/28S rRNA. So we are targeting with our kit a common sequence in this wide range with the 23S RNA and the 25 and 28S RNA. At all, we can say that we have no cross-reactivity with human genetic materials or common viruses. Here you can see a table and the results of an in silico test based on the SILVER database, which contains high quality and extensive ribosomal sequence data. From this data here, we can say that our design primer and probes are targeting highly conserved regions, and that our primers and probes are not affected by RNA secondary structures. But all in all, we have to say in silico prediction do not always ensure successful PCR amplifications. So it's just a prediction here. The third point is our optimized lysis method. You can see here the variation in the composition of different cell walls from gram-negative bacteria, gram-positive bacteria, fungi, and yeasts. Because of the different variations in the composition of the cell walls, we developed an optimized lysis method with multiple enzymes and detergents to detect all of them in one assay. The first point is the pre-incubation step that was included from our side, and it's regarding the condition of the standard strain. So normally you have the bacteria in the wet or growth phase. That means you have a lot of RNA available, and this is very easy for the NAT detection. On the other side, you can have the bacteria in the dry or freeze and dry state. So that means they are in the spore and dormant state. For example, the product BioBo from bioMérieux in this state. And it means you have a smaller amount of RNA in comparison to the wet one, and you have a spore hard shell around the bacteria, so it's very difficult for the NAT detection. What we developed with our Ribonat kit is a pre-incubation step to activate these microorganisms before, so that you can handle the wet and dry bacteria, or the state of the wet and dry bacteria, both with our kit. So let's come to the last point, the reduction of false positives. So firstly, our reagents are treated during the production in a way that all remaining nucleic acids are completely removed. You can see here a picture or a fixture before the treatment and a fixture after the treatment. And what we can see is that after the treatment, there are no false positive results. Then we have the inactivation of the residual DNA. That means that free DNA can no longer be amplified using the PCR. And our last point for the reduction of false positives is we have a two-stage DNA degradation treatment included of intermediate and final solutions, which reduces the DNA contamination during our whole process. With this, I would like to come to the outline and flow of our assay. So I told you before that we have 3 different kits that you need for one assay, and you can see here all the components that are included in each kit. All in all, we have to say that you need all 3 kits for our assay. So it's not possible to buy only one kit, you need really all 3. The required instruments you need is a benchtop centrifuge, a block heater for different tube sizes, a vortex mixer, a magnet stand, a centrifuge, and a 1.5 ml tube rotor, a thermostatic chamber to heat up to 60 degrees, and a real-time PCR system. In addition, you need different pipettes and consumables, pipette in different sizes from 1 microliter to 5 ml. You need an electronic pipette, an electronic multi-dispenser pipette, a real-time PCR plate and the plate seals, bigger tubes with 15 ml, and different pipette tips for the different sizes of the pipettes. For your working places and your working clothes, you would need a disposable cap, eyeglasses, a disposable mask, disposable gloves, and a disposable lab coat. All procedures should be conducted in a safety cabinet, and before bringing any equipment or reagent tubes into the safety cabinet, they should be thoroughly wiped with hypochlorous acid solutions. So all in all, we can say you need an aseptic environment and aseptic techniques to conduct our assay here. I showed you the procedure of our assay before, but now I would like to go into more detail on each step and explain each step thoroughly for you. So let's start with the first step, the pre-treatment. You have your sample, and your sample is at first splitted into 2 parts with 1 milliliter. One part is used for the aerobic bacteria and the fungi, and the other one for anaerobic bacteria. Both samples are centrifuged to form a pellet and then treated with different solutions. Different solutions because you have the 2 different samples here, one for aerobic again and one for anaerobic again. These mixtures are then incubated at 37 degrees for 3 hours and then centrifuged again. Finally, both treated samples are then combined again into one tube. And this pre-treatment step is critical to activate the microorganisms while inactivating the residual DNA. The next step is the RNA isolation. First, an enzyme enhancer is added and the sample is then centrifuged again. After that, the supernatant is removed and an enzyme mix, protein kinase K solution, a lysis buffer, and a lytic enhancer are added to break down the cells and release at this stage the RNA. After an incubation step, the internal control RNA, the binding buffer, the magnetic beads are added to the solution, and it's again incubated. The step is that the magnetic beads can bind to the RNA for isolating it easily. Following this step, the DNA's mixture is added and a washing buffer to remove any remaining DNA contaminants at first, and to clean our RNA. Then, finally, an elution buffer is added to release the purified RNA from the magnetic beads, and the eluted RNA is transferred to a new tube here at this stage. In the last step, the measurement follows. So at first, our RNA solution is again treated with a DNA solution and a second washing buffer, and again incubated to remove again any remaining DNA you could have in your sample. In the next step, the final processed RNA solution is mixed on the PCR plate with the detection mix. In the detection mix, you have different primers and probes, one's for bacteria, one's for fungi, and one's for the internal control, because later on, all of them have different wavelengths for the detection. The plate is then placed into the real-time PCR system for the one-step reverse transcription process, and the fluorescence is measured at this stage. So let's come to some experimental data for our kit. And I would like to start with the sensitivity here. What you did here is we detected 6 species, which are described in the pharmacopoeia, and spiked our samples with a concentration of 9 colony-forming units per ml of them. You can see all the 6 different species here. The CT value means the cycle threshold value is specified in each diagram. And this value indicates the number of PCR cycles after which the fluorescence signal exceeds the defined threshold. So that means lower CT values indicate an earlier and therefore stronger amplification. And what we can see here on this slide is that our kit was able to detect at a concentration of 9 CFU per ml all of the 6 species which were described in the pharmacopoeia. Then we did also the same with a more complex matrix with cell suspension samples and used therefore HEC 293 cells. We spiked here again with a concentration of 9 CFU per ml, and we can say also here that our kit is able to detect at a concentration of 9 CFU per ml all 6 species described in the pharmacopoeias. We did the same with mesenchymal stem cells and T cells, and also there the 9 CFU per ml were detectable with our kit. Then we did also a higher sensitivity method. That means we spiked here with a concentration of 2 CFU per ml, but we changed the incubation time. So our standard incubation time is 3 hours. We prolonged it to 6 hours, 13 hours, 14 hours, and 15 hours, and used the 3 species mentioned below. And what we can see here is that our extended incubation time enhances the sensitivity for each species, and that it was able to detect also in a concentration of 2 CFU per ml strictly anaerobic bacteria and slow-growing bacteria when we used a time or incubation time of 14 hours. Then I would like to show you some data from the reduction of the false positives. In the first picture, you can see the result for a commercial DNA kit, so for DNA extraction and detection, and below the same for our RiboNut kit. You can see the threshold line, and what we can see here is that you have a lot of false positives results for all 6 species for the DNA extraction and detection kit, but for our RiboNut, we have no species which were over the cutoff value, so no signal reached the threshold line. So we can say our RiboNut kit can reduce false positives derived from the residual DNA. Then I would like to show you some validation data for our kit, and I would like to start with the accuracy here. So a starter mixture containing 550 CFU is used from the product BioBall from bioMérieux. And this concentration is diluted in several steps with the Ringer solution to have at the end 4 different microbial concentrations with 18 CFU per ml, 9 CFU per ml, 4.5 CFU per ml, and 2.25 CFU per ml. For each concentration, both compendial methods are carried out, so one with the soybean casein digest medium and one with the thioglycolate medium. And on the other side, we tested the same samples with our RiboNut kit with the standard method and an incubation time of 3 hours, and with a high sensitivity method and an incubation time of 16 hours. And we can see the results here. So with the compendial method, we saw that all 6 strains could be detected at a concentration of 2.25 CFU per ml. For our RiboNut kit, we have to distinguish between the 3 hours incubation and 16 hours incubation. And for this 3 hours incubation, all 6 strains were detected at a concentration of 9 CFU per ml. 2 were detected at a concentration of 2.25 CFU per ml. But for the 16 hours incubation, so the prolonged incubation time, the kit detected all 6 strains at a concentration of 2.2.5. Let's come to the limit of detection. The number of assay was here 24. One assay means one RNA extraction, but we used this extraction for 2 wells. So all in all, we have 48 wells. And the LOD for our RiboNOT standard protocol, 3 hours incubation for the 6 microorganisms, was here 9 CFU per ml. For 2 special types of bacteria, for the Bacteroides fragilis and Cutibacterium acnes, we received an LOD of 36 and 99 CFU per ml. We did the same here again for the cell suspension samples and can see that we have exactly the same results as for the cell-free samples. So again, an LOD of 9 CFU per ml for the 6 species mentioned in the pharmacopoeia, and for the 2 special ones, 36 and 99 CFU per ml. Then I want to show you some results regarding the specificity in culture media and solutions. We used here some examples. The Ringer's solution as a representative example of cell washing solutions, DMEM as a representative of basic culture media, then a special culture media for MSC cells and a special culture medium for T cells. And you can see the results here. You have 3 different columns for the internal control for bacteria and fungi, so 3 different dyes. And we can say here we have no cross-reactivity for each of them and no false positive results. Also here we did the same with cell suspension samples and used mesenchymal stem cells, T cells, and again the HEC 293 cells. And also here we can say the same. So we have no cross-reactivity regarding the specificity results here. Then I want to show you the robustness of our kit with the cell suspension samples. Therefore, the samples were also spiked with a concentration of 9 CFU per ml with different bacteria and fungi. The tested cells are again mesenchymal stem cells and T cells, and you can see the 3 different kinds of bacteria and fungi tested here in the last point. And the bar chart shows here the CT values for each microorganism and each cell types. The individual points represent the values of different repetitions from 1 to 6, which are grouped close to the average value. And what we can say from this slide is that the CT values are consistent across different replicates and cell types, indicating a stable and robust performance of our RiboNOT kit. And last but not least, I would like to show you the ruggedness. Here, 3 different operators conducted 8 assays using different product lots. And the test here was also conducted using the 6 species mentioned in the pharmacopoeia with the same concentration as before. And we can see here the results for the different species. So we received also here consistent CT values for all results and across all operators for all 6 species and for different lots. So with this, I would like to thank you. That was all from my side. We can summarize that our kit performs very well in all the experimental data and validation data. And with this, I would like to thank you for your kind attention, and I would be happy to answer any questions you have regarding our RiboNOT kit.
Perfect. Thank you, Dr. Matros, for that fantastic presentation. So let's move on to the final part of today's webinar, which is the Q&A session. So the first question that we've got is, what makes RNA-based detection more effective in identifying live microorganisms compared to DNA-based approaches?
Yes. So our RNA-based detection method offers several key advantages for the detection of the microorganisms, and I would like to mention 4 of them. So at first, we have an indication of microbial viability. That means the ribosomal RNA molecules are present in high copy numbers only in metabolically active cells and degrade rapidly after the cell death. In contrast to the other kits, which are detecting DNA molecules, they can persist a long time after the microorganism have already died, which can lead to false positive results. Then we have, in general, a higher sensitivity because we are targeting the ribosomal RNA instead of the DNA. I showed it in the presentation before. We have a less interference from dead cells, and we have a rapid response because our ribosomal RNA detection level correlates tightly with the cellular activity. So all in all, we can say we have 4 key points why it's more effective than detecting the DNA.
Okay, that's great, thank you. And the next question is the method suitable for all product types, or are there any limitations?
Yes, so our method, our kit, is highly versatile and applicable to different sterile pharmaceutical products, including, for example, cell-based therapies and short shelf life products. It is not clear yet whether there are other restrictions, but what we can say now is you have to be careful when using samples containing antibiotics, because there may be an effect on the bacterial activation stage.
Perfect, thank you. So the next question we've got is, what types of microbial contaminants can be detected using this approach, and how comprehensive is its detection range?
Yes, so we are detecting a wide range of microbial contaminants, including gram-positive and gram-negative bacteria, as we are detecting the 23 sRNA sequence. We can target the fungi and yeasts because of the 25/28 sRNA. So all in all, we can say that we are covering a wide range of microorganisms.
Perfect, thank you. And the next question we've got is additional equipment or infrastructure required to integrate this method into existing lab setups?
Yes, so no, our method is designed to be compatible with standard biological laboratories that you can find in all QC departments. However, you need, of course, some machines or have to implement some things to conduct our assay. For example, you need the real-time PCR instrument. You need some sample preparation tools. I showed it in my presentation. For example, a vortex mixer is centrifuge, but normally they are already available in biological laboratories. You need a contamination control measurement because of the high sensitivity of our product, and it would be good to train your personnel regarding the PCR. But all in all, the most of the things should be available in the QC departments.
That's great, thank you. So we've got time for one more question, and this one is, what training or expertise is needed for lab personnel to adopt this advanced testing technology?
So normally your personnel should have the basic molecular biological skills. So they should be familiar with nucleic acids, etc., and the amplification procedure for the PCR. They should have an understanding of the aseptic techniques to work in an aseptic environment. And they should have, of course, a detailed look to our protocol, to the different steps that have to be conducted. And they should be able to perform some troubleshooting if you would see problems during the procedure. But all in all, basic molecular skills should be enough to conduct the assay.
That's great. Thank you so much, Julia. Well, that's all we've got time for today. If we didn't get around to your question, we will follow up with you after the webinar. So first, a huge thank you to our expert, Julia, for a fantastic presentation. And thank you to everyone joining us online and for also submitting your questions. This webinar will be available to watch on demand. So as you leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, it will be sent to you via email shortly, and we would greatly appreciate your feedback. So on behalf of European Pharmaceutical Review and Fujifilm, thank you all for attending, and we hope to see you again very soon. Goodbye, and thank you for joining us.